Hello everyone. Welcome to our educational webinar titled From Stem Cells to Organoids, Modeling the Gastrointestinal Tract. My name is Jenny Marie Petersland and I'm a Product Marketing Manager at Stem Cell Technologies, and I'll be your moderator for this webinar. And it's now my pleasure to introduce you to our speaker today, Dr. Meritxell Huch. Dr. Huch is a junior group leader at the Gurdon Institute, University of Cambridge in the UK. Prior to joining the Gurdon Institute, Meritxell completed her postdoctoral research training in Dr. Hans Klever's lab at the Utrecht Institute for Developmental Biology and Stem Cell Research in Utrecht, the Netherlands. And here she isolated gastric stem cells and demonstrated their ability to generate organoid structures. She then later studied hepatic and pancreatic progenitor cells for regeneration and cell therapy applications, and discovered a method for their long-term in vitro expansion. Meritxell's current research uses the liver and pancreas as models for investigating the mechanism for adult tissue regeneration. And Meritxell has a PhD in gene therapy and pancreatic cancer from the Center for Genomic Regulation in Barcelona, Spain. Let me now hand you over to Meritxell to begin the presentation.
Hi, everyone. First of all, thank you very much, Jenny Marie, for inviting me to give this webinar here today for Stem Cell Technologies. It's my pleasure to try to explain you how, during my postdoc and in my new lab now, how the study of adult stem cells brought us to identify a way to grow these cells in culture in what we call organoid cultures. And what I'm going to explain you today is how we use these organoids to model gastrointestinal tissue. So, as you very well know, the intestine is an organ that is very suitable to the study of stem cell biology, because in the intestine, proliferation and differentiation, they happen at the same time, at the same moment. In fact, it was very well known that proliferation was driven by stem cells located at the base of the intestinal crypt. The intestinal crypt is this invagination of the mucosa into the submucosa. So it was well known that actually stem cells were located at this base of the intestinal crypt, and they would be proliferating and giving rise to daughter cells that will be pushed upward into the crypt-villus unit. And once they would reach the crypt-villus axis, they will start differentiating into the cell types, into the enterocytes and goblet cells, which are the cells present in the intestinal tract and are responsible for the function of the intestine. So when Nick Barker was studying the Wnt signaling pathway in the intestine, he identified this marker, which is a Wnt target gene LGR5. He identified that this LGR5 protein, a 7-transmembrane domain protein, was marking exclusively a subset of cells at the very bottom of the intestinal crypts. This had been previously described as the crypt-based columnar cells. And LeBlanc, in the mid-50s, he had postulated that these cells might be stem cells in the gut because they were actively cycling. So at that moment, there was not the technology, but Nick Barker, when he identified this LGR5 as a marker of this subset of cells, he asked the question, are these stem cells in the gut? And to try to answer that question, he utilized the Krylov-Spitz technology that is very well known now in the stem cell field, with the advantage of trying to trace the fate of these LGR5 positive cells in vivo in real time. So what he did, Nick performed what we call lineage tracing experiment by combining this LGR5 GFP-Cre with a ROSA26 reporter. And in that case, he utilized the LACZ reporter. And for those of you that you are not familiar with the system, in that case, when we have a mouse that in the LGR5 allele, there is the Cre, inducible Cre, and in the ROSA locus, you have a reporter, in that case, the LACZ. When we utilize the Soriano reporter, that there is a stop codon in front of the LACZ, these cells that express LGR5, they do not express the LACZ. However, if we give tamoxifen to these cells, the CRE becomes activated, it goes to the nucleus, it will eliminate this stop codon in front of the laxer reporter, and that will allow that these LGR5 positive cells can be now marked by the expression of the ROSA locus. In that case, it was marked with the laxer reporter. So they were marked in blue. So utilizing this technology, Nick Barker identified that these LGR5 positive cells, these crypt-based columnar cells located at the very bottom of the intestinal crypt, they were indeed true stem cells in the gut because they were self-renewing, they were proliferating at the same time that they were generating all the progeny, so also differentiating into the intestinal cells. Of course, LGR5 is a marker of stem cells in the gut, but that doesn't mean it's the only marker. There are other markers, and also other cells have been identified as a potential stem cells in the gut. And this is exemplified in this slide, where what we see is that the stem cell zone, we can identify these LGR5 positive cells. They will give rise to entire crypt villus units, indicating that they self-renew and differentiate into the intestinal epithelium. And also, later on, and previously as well, other cell types, in that case, marked either by delta-like 1 or label-retaining cells or marked by other markers, other cell types have been also identified that in the case where LGR5 positive cells are damaged and are gone from the epithelium, these cells can revert back and they go out from quiescence and they acquire a stem cell fate in order to, again, maintain intestinal homeostasis. In a follow-up study, it was very interesting, of course, that LGR5 positive cells were actively cycling. So in a follow-up study, Toshisato in the lab identified the way to maintain the self-renewing status of these cells in vitro in culture. In that case, Toshi in the lab isolated these LGR5 positive cells and embed them in a 3D matrigel in a matrix that would allow the cells to have a 3D dimension. And when he combined matrigel with this cocktail of growth factors, which included EGF, arspondin, or noggin, this combination of growth factors combined with the matrigel allowed that these LGR5 positive cells isolated from intestinal crypts, they could start proliferating in vitro in the same way that they would do in vivo. They would generate what we call these intestinal organoids or spheroids. And these cells are only generating the epithelium. There is no mesenchymal attached. This epithelium, thanks to the growth factors that we give on the medium, it self-organizes to try to generate these structures where we can see a crypt domain that is surrounding a central villus domain. And if we look at and the most interesting of this system is that it maintains the stem cell properties. In one hand, they self-renew so that we can maintain them as much as we want in culture. We have managed to split these cultures and from one well generate many, many wells in vitro for more than a year. But not only they self-renew, but they also differentiate into the cell types present in the intestinal epithelium. So what we see here is that Toshisato demonstrated that indeed these intestinal organoids, they retain the physiology of the intestinal crypt in the sense that they also contain LGR5 positive cells at the very bottom of the intestinal crypt. And on the other hand, they also differentiate in what we call in the villus domain. We also identify all the cells present in the intestinal epithelium. So Toshisato had identified a way to isolate these cells and maintain their stemness in culture, maintain their self-renewing and differentiation properties by culturing them in matrigel and with these growth factors. Therefore, the follow-up study, what was very obvious, was to ask the question, if these intestinal organoids are truly functional. One of the gold standard experiments to answer that question is to perform a transplantation assay. And for that, we collaborated with Mamoru Watanabe's lab to try to transplant intestinal organoids into, in that case, it was a mouse model with enterocolitis. So Toshisato, in the lab, he grew these intestinal organoids, and for that, we utilized a red mouse. So we generated red intestinal organoids that were transplanted into a mice with enterocolitis, which of course it was not red. The transplantation went as follows, and this is just an animation to try to illustrate how the transplantation worked. What we did, we collected as many intestinal organoids as possible, and via enema, these organoids were transplanted into the anus of these mice that were harboring intestinal lesions done with this DSS, which is a model for enterocolitis. And what we observed is that these intestinal organoids, they found the lesions and they engrafted into these lesions, trying to repopulate the damaged areas. And this is an illustration or animation of how the process was going on. And what we observe here is the data that we obtained from these mice. So actually, 3 months after the transplantation, these red colonic crypts that we find here, these red colonic crypts, were found in the intestinal epithelium of these mice, indicating that these cells had not only engrafted but also participated in the normal homeostasis of the tissue. Of course, these colonic crypts also contain all the enterocytes and colonic epithelial cells classical of the colonic crypts. I want to mention here very clear that indeed this had engrafted and maintained and persisted for these 3 months in culture, and we have to remember that the intestinal epithelium is renewed every single week. Because this was identified 3 months later, this was indicating that indeed these intestinal organoids had engrafted and contributed to the normal homeostasis of the tissue. In parallel to all these studies, Nick Barker and myself, we were interested in looking at the stomach epithelium. Because indeed, Nick Barker, when I joined the lab, he also had shown that LGR5 was also a stem cell in the stomach epithelium, particularly in the pyloric epithelium, or the antropyloric region of the stomach, which is the region closest to the duodenum. In that antropyloric region of the stomach, LGR5 positive cells contribute to the normal homeostasis of the stomach epithelium, and indeed they again maintain these self-renewing and differentiation properties classical of the stem cell. They will generate these very nice gastric glands containing the different gastric epithelial cells. So what I asked the question was whether if we isolate these LGR5 positive cells, can we also maintain their stiffness in vitro in culture? And after modifying the culture conditions for the small intestine, it was very clear that an FGF signaling as well as Wnt signaling were essential for the self-renewing and maintenance of these gastric stem cells in culture. In fact, we identified that FGF was essential mainly because actually there were very nice studies on the chick embryo that were telling us that FGF is a requirement to maintain stemness in the stomach. So addition of FGF10 in combination with Wnt signaling facilitated that these stomach stem cells now, they would start proliferating, they would generate this, what we call now stomach organoids. Again, this is not containing any mesenchymal tissue, so maybe the word organoid is not completely correct. We should maybe utilize spheroid, but for the time being, I'm going to call them stomach organoids because this is how we've been calling them in the lab. And actually, what we observed very clear is that these organoids will very quickly generate these very nice budding domains that are surrounding here, and that they very much resemble to the stomach epithelium in culture. In fact, in this very nice magnification, we clearly see this glandular-like domain surrounding the central villus domain, the central lumen. And when we looked at the expression pattern, we did observe the appearance of all the different cell types present in the stomach epithelium, as well as the expression of LGR5 located at the very bottom of these stomach glands, as it was in vitro. In vitro, it was modeling what it was in vivo in the mouse. So, as a summary of this first part, just to remind you that in the lab, thanks to the study of the signaling pathways involved in proliferation and differentiation, Nick Parker identified LGR5 as a stem cell marker in the gut and in the stomach, and knowing the signalings that would maintain stemness allowed us to transfer the ability of these cells that would renew in vivo to transfer these in vitro into what we call these organoid stem cell cultures that recapitulate intestine and stomach epithelium in vitro in culture. So the next question was very clear. So what happens to all these other organs that do not proliferate extensively, like the intestine or the stomach? What happens to organs like the liver, lung, or pancreas, which they do not proliferate? Can we also generate models in vitro for study either development or disease mechanisms? And there is different ways. The whole stem cell field is trying to identify these ways of trying to transfer how to grow organs in culture. And for that, we can choose different strategies. We can choose to start with embryonic stem cells or induced pluripotent stem cells or perinatal cells, or we can also choose to start with adult stem cells. Of course, for those organs where these either have been identified or have been hypothesized to exist. And here we have a summary of most of these papers that have been breakthroughs in the field, from being able to grow cerebral organoids from the Novlik lab, to grow, of course, optic cup structures from Sasai's lab, or in our case as well, we've centered these studies have all been centered in starting from embryonic stem cells or iPS cells, or in the case of Angra Pemberton, she also started with embryonic pancreas and constructed a very nice pancreas development culture in vitro. In our case, I'm going to center my talk today on what we described as an organoid cultures starting from adult tissues. And I'm going to center that into liver and pancreas. As most of you will know, the liver is one of the major is actually the biggest internal organ in our body. And it's mainly formed by these cell types that we call hepatocytes, which are the true functional cells of the liver. They secrete albumin, they detoxify from drugs, they produce bile acid, and these hepatocytes are surrounding this network of the biliary ducts that are formed by these ductal cells. Hepatocytes and ductal cells are the epithelial cell types and are endodermal derived cell types of the liver, while the other cell types, they work in conjunction with the other cell types that are not endodermal derived, like the Kupffer cells or stellate cells. The liver is a very interesting organ because it has a huge regeneration capacity after damage, and actually it is very well described that upon partial hepatectomy or upon toxic injury that causes up to 70% of damage into the liver, for all the vertebrates studied, usually adult cells will generate adult cells with little contribution from stem cell compartment. However, in many of the vertebrates studied, when there is a huge damage into the hepatocyte compartment, including up to 90% or 95% of hepatocyte loss, it has been described, for instance, for the zebrafish, that ductal cells can then differentiate into a progenitor state that will afterwards differentiate into hepatocytes and ductal cells again. And the same happens in fulminant liver failure, where if you have a 90% hepatocyte loss in a fulminant liver failure, however, 30% of these patients will recover, and if you take a biopsy of these patients during the moment of the failure of the liver, what you would see is a huge proliferation of small cells that have been called oval cells, progenitor cells, or dedifferentiated cells. It is not known where they are originating from in the human yet, but it is clear that 30% of these patients recover in a week, indicating that probably these cells, despite it's not fully proven, but is an interesting indication to say that these cells probably differentiate into the functional cells of the liver. So, because we were studying LGR5 as a win target gene, we also, of course, were interested in looking in the liver. And when we did that, what we observed, utilizing again lineage tracing experiments, is that if we damage the liver and we now induce lineage tracing, what we see is that LGR5 positive cells will contribute to the generation of hepatocytes as well as ductal cells. But of course, this experiment cannot rule out whether these ducts and hepatocytes that are labeled here in this picture, whether they come from the same cell or actually there is two independent LGR5 progenitors that will give rise to both hepatocytes and ductal cells. And we are trying to elucidate this question. But what was very interesting from this experiment, it was the fact that actually we had a marker to identify the cells that are proliferating. So I asked the question whether if we isolate these LGR5 positive cells, can we grow them in culture? Can we keep their proliferation status that they have in vivo during the regenerative response? Can we keep it in vitro in culture? And of course, when we put these cells in combination with the growth factors like HGF and FGF, Wnt and nicotinamide, what we observed was that these cells will start proliferating. They will generate these, again, 3D structures. This is again in Meritxell, and they generate these 3D structures that are surrounding the central lumen. We can expand them in vitro, in culture, as much as we want by splitting them and generating from one well, generating 5 to 6 wells every week for more than a year. So what we asked here is, can we do the same with a normal liver? And the answer was yes. Actually, if we isolate ductal cells from a normal liver, and probably the isolation procedure is what is causing the damage to the liver, this is triggering a regenerational response in a way that these nice isolated ducts will directly start forming, close very quickly, will directly start forming these, again, organoid structures. And if we profile these organoids generated from the duct structures as well as the organoids generated from the LGR5 positive cells, what we observe is that actually they look exactly the same. Gene expression is identical, and what it happens is that we've induced this stem progenitor state in vitro in culture. The next question, of course, was what are these cultures? And as I mentioned already, these cultures express stem cell markers, but also mainly they are ductal-like structures. As you can see here, this is a single layer epithelium of columnar cells that are positive for the duct marker keratin 19. And when we transfer these cultures into a differentiation medium in which we inhibit Notch and TGF-beta signaling, what we observe is that this columnar epithelium changes into a pseudostratified epithelium, and the cells are not columnar anymore, but they show this very nice polygonal shape, and they start expressing the markers of the hepatocyte lineage. Even more interesting, what we see here is that some of the cells even acquire a binucleated phase, which is a mature final fate of hepatocytes. And I will show you later that we also performed some in vitro studies, and we also observed that they maintain their functional characteristics of liver cells in vitro. But to fully prove that these cultures that had been differentiated into hepatocyte-like fate were indeed functional, we performed an in vivo experiment. And for that in vivo experiment, we collaborated with Craig Dorrell and Marcus Grompe at the OHSU, Oregon, in Portland, Oregon. And we collaborated with them because they had this very beautiful mouse model, FAH knockout mouse model, which is a model of tyrosinemia type 1. In that model, the mice is lacking FAH, and because of that, tyrosine cannot be metabolized into its normal pathway, and it happens that it accumulates these metabolites, intermediate metabolites, that would result is that actually these mice, as well as the humans affected by this disease, they undergo liver failure very quickly postnatally. However, it was identified that NTBC is a drug that prevents that tyrosine to enter this pathway, and it allows the mice as well as the humans to be maintained alive at least for a certain period of time. This model, it was optimal for us to be able to ask the question if our organoids indeed were capable of generating functional cells in vivo. For that, we isolated LGR5 positive cells. We also did that with normal organoids, isolated from ducts, generated organoids, differentiate them, and transplant them into these FAH mutant mice. And then, after transplantation, we removed the drug and asked the question, can the hepatocyte-like cells from the organoids become fully mature hepatocytes into the livers of these FAH mice? And if so, are they secreting FAH? And if so, do they rescue the phenotype of the mice? And what we observed is 3 months after the transplantation, 25% of our mice had received cells. We could detect grafts that were around 1% of the liver tissue. And then we asked the question, is this graft functional? And of course, with 1% of liver tissue, we could not rescue the phenotype of these mice. But it allowed us to answer the question whether these cells were or not functional. And what we saw is that indeed, those mice that had received grafts, and that had received cells, and we could detect grafts, were surviving longer than those same mice that had received the same cells, but we could not detect grafts. And the survival was even more significant when we were comparing to the mice that didn't receive any cells, indicating that indeed these grafts were secreting at least some FAH protein that would allow the increase on the survival rate of these mice. So, of course, this system is very interesting, and it allows us to ask many questions regarding mouse liver biology. But if we cannot transfer this technology to the study of the human setting of the humans, the real importance of the system is very poor. So, we got into the enterprise of trying to transfer this technology into the human setting to try to study human development and human biology. And for that, I have to thank exclusively, I have to thank strongly Luc Vandelaan and Monique Verstegen in the Erasmus Rotterdam Center, because we got, thanks to them, we got enough liver biopsies, human liver biopsies, to be able to perform the following experiments. So the experiments that we designed were as follows. We obtained liver biopsies from donor material. From these donors are patients that die during a car accident. They don't have any disease. Then they donate their organs, and before the organ is being transplanted, we get a biopsy of this liver, and then we put it in culture and we try to establish organoids from this. And of course, it took a while to identify the culture conditions because the mouse media for mouse liver organoids didn't allow the expansion of these cultures. That's why we combined the mouse medium with TGF-beta inhibition and cyclic IMP activation. We obtain a culture system, very robust culture system that would allow the long-term expansion of human liver biopsies in culture. Here we can see the expansion potential of these cultures is huge, so they are growing exponentially for at least 4 to 6 months that is when we stop these experiments. And then the question was, we now can expand these cultures as much as we want, and can we, what is the cell of origin? What is the cell of origin that starts these cultures? And then what we observed is that EpCAM positive cells, which are keratin 19 positive, can establish organoid cultures in vitro. As we can see, one of each of these structures is an organoid, and the efficiency here it was very high. It had a 30% colony formation efficiency from EpCAM positive cells derived from these human livers, and whereas EpCAM negative populations did not start organoid cultures, at least from the humans. So what we had in culture, it was a population that was starting from EpCAM positive and was being able to be maintained in vitro in culture. The characteristic, the ability to be able to sort out these cells allowed us to actually start organoids from single cells. And these clonogenic cultures allowed us to ask the next question, which indeed was, are these cultures genetically stable? Because of course, if an organoid culture is not genetically stable, not an organoid culture, I mean any type of culture, if it's not genetically stable, then its utility to being used, for instance, for regenerative medicine or disease modeling drops very dramatically because it becomes an unstable culture. So that was very important for us to try to know whether these cultures were genetically stable, and for that we performed the following experiment. What we did here was, again, isolate the liver biopsy, and thanks to Edwin Kuppen and Ruben van Boxtel at the Hubrecht Institute, we could perform whole genome sequencing of these cultures and ask and answer this question. And I will just go with you through the experiment, because what we did was we got these liver biopsies, we collected some part and keep it for whole genome sequencing, we culture them for vid, and then perform clonal cultures, so started from one single cell. We expanded these clonal cultures to obtain enough material for collecting for whole genome sequencing, and then we kept these cultures expanding for more than 3 months when we subcloned them, and again we expanded to get enough material for whole genome sequencing. And here we compare the subclones to the clones to the original biopsy. The comparison of the subclones against the clones gave us the somatic variation after long-term culture. And what we observed is that indeed in coding regions there were no base substitution. We could not identify any base substitution in any of the clones, which was at least 3 subclones per clone for the donor 1, neither for the donor 2. We only find 1 signal mismutation in one of the subclones of the donor 2, indicating that long-term expansion indeed results in no accumulation of somatic variations or in coding regions. Of course, we identified mutations in non-coding regions. And more interesting, when we looked at the comparison of the clones versus the original biopsy, we again indeed, summing all the clones that we had obtained, we observed 16 and 17 coding mutations in these clones. Now we don't know whether these 16 mutations accumulated in the different clones, summing the different clones, were present already in the liver biopsy or actually well generated during the derivation process, because the liver biopsy, of course, was not sequenced at the same clonal level. But we are trying to investigate that at the moment. So now we had a culture that it was genetically stable, that we can long-term expand from human liver biopsies. So then the question is, what are these cultures again? And what we observed is that they express stem cell markers as well as they express ductal markers. So again, they resemble very much to the mouse scenario. Now the cultures again are, if we look for the ductal expression, we observe that they are single-layer epithelium of columnar cells expressing this very nice keratin 19 staining. And when we differentiate them, again, removing the growth stimuli and inhibiting Notch, inhibiting TGF-beta signaling, we observe that indeed this columnar epithelium again changes to this octagonal or polygonal shape classical of hepatocytes and start expressing classical markers of hepatocytes like albumin or ZO1. Interestingly, we performed a gene expression analysis and we observed that albumin was slightly lower, like 100 to 1,000-fold lower gene expression. However, cytochrome was almost equal to the same expression that you see cytochrome in the liver tissue. And more interesting, when we perform functional assays in vitro, we asked the question, do they secrete albumin? And the answer was yes. The organoids, after differentiation, they secrete albumin, and we have hepatocytes here, fresh isolated human hepatocytes. We would see again this 100 to 1,000-fold less secretion, and it's clear that this secretion is going to be less because if we look in this picture, we already see that not all the cells start expressing albumin. So there is still room for improvement on these differentiation protocols to achieve a much more efficient differentiation in vitro. Nevertheless, when we looked at the function of the other functional assays, we observed that cytochrome activity was again very similar to what we will have in fresh isolated hepatocytes, and that are not plotted in this graph because it was not done in the same experiment, and they also, for instance, could metabolize some drugs like midazolam. And again, similarly to what we had done in the mouse, we performed a transplantation assay to ask the question whether these cells that had differentiated in vitro were capable of fully maturated in vivo in culture. And what we observed is that indeed there were human albumin positive cells in these mice, immunosuppressed mice, which we had induced liver damage using CCL4 retrosin, indicating that the human liver organoids pre-differentiated in vitro had engrafted and differentiated into human albumin positive cells, lost their keratin 19 staining, and keep secreting human albumin for the whole time of the experiment. So then here we had now a system where we could not only expand human liver cells for a very long period of time, and that these cells were genetically stable, but they would also differentiate into a functional in vitro culture system, into a functional hepatocyte-like cells in vitro. And then the question was, can we use this system to model some human liver diseases? And for that, we chose alpha-1 antitrypsin deficiency, which is a rare disease, but is a monogenic disease that affects some population because they are deficient in this alpha-1 antitrypsin protein. And alpha-1 antitrypsin is a monogenic disease with which several alleles have been described, but the most interesting allele for us was the ZZ mutant allele. And we were lucky enough to, through our collaborator Luc van der Laan at the Erasmus Rotterdam Center, we managed to obtain biopsies from 3 patients harboring this ZZ allele. And this allele is interesting for us because in alpha-1 antitrypsin deficiency, the patients mainly have a lung disease. So the alpha-1 antitrypsin deficiency is a lung disease. But it's a lung disease that is generated on the liver. And the patients harboring the ZZ allele, which is the most common, they not only have a lung disease, but they also have a liver disease. And this is because in this particular case, alpha-1 antitrypsin protein changes, gets misfolded, and then it makes these polymers that will precipitate into the cell. And actually, if we look at histology of a patient, it's exactly what we see in this image. What we see is that in a normal patient, in a normal donor, someone that doesn't have alpha-1 antitrypsin deficiency, alpha-1 antitrypsin is produced in the liver, it's released into the blood vessels, into the circulation, and it goes into the lungs, where it will protect the lungs from being lysed by the neutrophils. So in a histology of a healthy liver, alpha-1 antitrypsin is not present. However, patients with alpha-1 antitrypsin deficiency, their livers cannot produce, they produce alpha-1 antitrypsin, they produce alpha-1 antitrypsin, but as you see in the image, the alpha-1 antitrypsin precipitates into the hepatocytes of these livers. And that has two consequences. One consequence is that alpha-1 antitrypsin is not released into the bloodstream, therefore these lungs are not protected and are being digested by the neutrophil elastase. But the second consequence is that these precipitates of this misfolded protein cause the death of these hepatocytes. And actually, these patients require a liver transplant not only to facilitate the release of alpha-1 antitrypsin in the bloodstream and alleviate the lung problem, but these patients also need a liver transplant because their livers are very unhealthy due to this constant damage caused by the precipitation of alpha-1 antitrypsin, as we see here in brown, in these hepatocytes. So then we ask the question, can we model this disease in vitro in culture? And because this is a disease of hepatocytes, we were lucky that progenitors are not affected, or ductal cells are not affected. So we isolated these liver biopsies and we put them in culture, and what we observed is that alpha-1 antitrypsin deficient patients biopsies of these patients could generate normal organoids. It's true that these organoids had a slightly different shape than normal organoids, but the self-renewing capacity, the expansion potential was maintained, and that allowed us to ask the question whether these cultures would mimic alpha-1 antitrypsin deficiency in vitro. For that, what we did was transfer these cultures that were expanding into a differentiation medium, and then we start looking at different parameters. We looked at alpha-1 antitrypsin secretion, and as expected, donors, derived organoids, donor-derived organoids will secrete alpha-1 antitrypsin into the medium. However, the 3 patients that we studied, none of them could secrete alpha-1 antitrypsin into the medium, and similarly, the 3 patients could also not inhibit elastase, while the donors could inhibit elastase in culture. More interesting, when we looked at the histology of these differentiated organoids, what we observed is that donor organoids, or organoids derived from a donor patient, they do not have any alpha-1 antitrypsin precipitate into the cells. However, organoids derived from alpha-1 antitrypsin patients, they show this precipitate into the hepatocyte-like cells after differentiation, indicating that these cultures were mimicking what we were seeing in vitro. They're mimicking in vitro what we see in the histology of the patient. And of course, alpha-1 antitrypsin is a disease that not only shows epithelial phenotype, but there is also this damage into the tissue causes a lot of inflammation, and the organoids are not mimicking that at the moment, but at least give a platform to be able to study whether we can identify some drugs that would alleviate the precipitation of this protein into the tissue, and therefore they give maybe some potential personalized medicine for these patients. And in the last minutes of my talk, I'm going just to show you how all this knowledge that we acquired on the liver, we also in parallel transfer it to pancreas, and how we established pancreas organoids as well. As you know, pancreas and the liver and the pancreas, developmentally, they have a very similar origin. They actually come from the same endodermal progenitors. However, during development, they acquire their own specific fates and they become completely different organs. Nevertheless, they retain the same structure. They retain having both organs have a ductal system that is released into the duodenum, and both organs have dysfunctional cells that are surrounding this ductal system. In the case of the pancreas, of course, it's the acinous cells. In the case of the liver, it's the hepatocytes. And in the case of the pancreas as well, we have the islets, which contribute to the glucose homeostasis. Because they were very similar morphologically, if we look at them like this, they look very similar. We decided to isolate ductal cells from the pancreas and put them in culture, and what we observed is that indeed they generate these ductal structures, these organoid 3D structures, that if we look at them, what we see is this budding domain surrounding a central lumen, and if we make a confocal section, what we observe is that indeed this is a single layer epithelium of ductal cells. Then we ask the question, can we differentiate these cells in culture into an endocrine phase? And in vitro, we've not managed to do that, but in vivo, we ask the question what would happen if we do it in vivo. So for that, Paola Bonfanti in Harry Hemberg's lab helped us very much in generating these in vivo experiments. What we did here, we first generated, or Paola first generated this morphogenetic assay where she dissociated embryonic pancreas and put it into the kidney capsule after dissociation, and she observed that when you do that, indeed, this embryonic pancreas in the kidney capsule of the inefficient mice, it keeps differentiating and it generates all the 3 cell types present in the pancreas epithelium: ductal cells, endocrine cells, acinous cells. So then it was clear that this dissociated embryonic pancreas contains all the instructions, or contains all the signaling, or maintains all the signaling to instruct the epithelium to differentiate into the mature cells. So we ask the question, what happens if now we mix this embryonic pancreas with our adult derived organoids? And for that we used GFP derived adult organoids. We mix them with this embryonic pancreas, and we ask the question, can our organoids differentiate in this permissive environment? And the answer was yes, they can. Indeed, what we found is these GFP positive cells, they start expressing synaptophysin or insulin, which is of course endocrine markers, and they also express, we also observed that they express glucagon and somatostatin, and indicate in a monohormonal way, indicating that the embryonic pancreas could instruct the adult ductal organoids to differentiate once they were in the right environment. In the last 1 minute of my presentation, what I'm going to show you is how we've used this technology to model pancreas cancer. And we were lucky that at the HUBRAC we could obtain material from Professor Hofferhaus at the UMC Utrecht, and this material was derived from pancreas cancer. And what we see here is that pancreas cancer is mainly pancreatic ductal adenocarcinoma, but there is other different types of pancreas cancer, and we were lucky enough to have different types of pancreas cancer. Here we have a classical pancreatic ductal adenocarcinoma. This is the classical histology. We have an IPMN, which is a mucinous type of pancreas cancer, and ampullary tumor. We obtained organoids from these human pancreas cancers, put them in culture, and Sylvia Boch, my friend, she recently published this data, and she showed that when we grow normal human donor cultures from a pancreas, what we obtain is, as expected, a single layer epithelium of ductal cells. However, when we grow pancreas ductal adenocarcinoma, we again obtain this not structured epithelium, classical of a panin. But if instead of pancreatic ductal adenocarcinoma, we grow IPMN, the histology of the organoid is very different, resembling the original tumor, similarly what happens if you grow an alveolary tumor. So with that, I arrived to the summary of my talk, and I just want to mention that, remind you that either starting from a damaged liver or a normal mouse liver or a normal human liver or a mouse pancreas, we can obtain organoids if we isolate this ductal population, and these are mainly ductal-like cells which express stem cell markers. And now, if we centered onto the liver, what we see is that if we differentiate them into cells, the cells will start expressing classical hepatocyte markers, and we can transplant these cells in vivo into a mouse model of a liver disease, and we can find back the cells that we transplanted. So we expect that if now we do the same with human donors, we can isolate these positive cells, expand them in culture, we prove that they are genetically stable, we can transplant them back into the mouse, but also when we start with a human patient instead of a donor, we can also model diseases, and I've shown you 2 examples on alpha-1 antitrypsin and pancreatic cancer. So along with the other types of organoid cultures that are being generated from embryonic stem cells or iPS cells, adult stem cells are also capable of mimicking not only the normal adult tissue homeostasis, but also the adult tissue in disease, and we hope it will help for personalized medicine. And with that, I'm arriving to the last slide of my talk, which I would definitively like to thank Hans Cleves at the Hubrecht Institute in the Netherlands, in Utrecht, for not only being a fantastic mentor, but I learned a great deal in his lab. I learned a lot, and I'm very, very grateful. All I've presented here has been done in his lab. And I would also like to thank the other people in the lab, especially I've already mentioned Silvia Fernandez-Bock. She was the person involved in the Pancreas Project and also in the Pancreas Cancer Project. Rob Fries and Helmut Gerhard, he performed these human liver transplantation experiments, and Karin Hammer, who was an excellent technician, she was very helpful on the establishment on the human liver cultures. And I've already mentioned the collaborators during my talk, especially Craig Dorerick and Marcus Grompe for the transplantation experiments of the mouse organoids, Luc Bandelan and Monique, Jan Eisermans in the Erasmus University, who provided all these human liver tissues to be able to perform this experiment, Ruben van Boxtel and Edwin Kuppen, who performed the genetic analysis of the human liver cultures, and Harry Heimberg and Paola Bonfanti in Brussels, who performed the pancreas transplantation experiment. And this is my lab, now located in Cambridge, and we are pursuing some of these experiments to bring them further. And I would like to thank my funding. And with that, I conclude my webinar, and I will be very happy to answer your questions. Thank you very much.
Thank you very much, Meritxell. That concludes the presentation segment of the webinar, and we'll now go to the question session that you've asked by writing the questions throughout the presentation. As a reminder, if you do have any other questions, you can use the chat box to the right and we'll pass them on to Meritxell. There are quite a lot of questions, so we'll see how much we have time for. First, there are some questions about the culture system, and we can start with this one. First of all, can you do? I'm actually going to combine a couple of questions here, Meritxell. But can you do large-scale culture, and can you cryopreserve the organoids so that you can kind of keep them as cell lines growing indefinitely?
Regarding the cryopreservation, which is the easy one, yes, the answer is yeah. For all of the cultures, we've managed to freeze and thaw them afterwards, and with complete preservation of the characteristics. And in that sense of the self-renewing and also differentiation capacity. And regarding the large-scale question, the truth is that with the largest scale we've ever done was to generate around 1 million cells, so 20 million cells for transplantation experiments, but larger than that we've not done. So everything has been done, so all these transplantation experiments we've done them manually, as I say, so no rotor or no suspension culture. So far this is not being done.
And when you say 20 million cells, how many mini-guts would that more or less average? Do you know?
That was, for us, that was around 6 plates of 6 well.
Yeah. Wow.
I don't know if that gives an impression, something like that.
Thank you. And then you use a defined system with specific growth factors. In your opinion, what are the benefits of using conditioned medium versus specific growth factors? And also, have you tried the new medium from stem cell called intestinal?
That's a good question. So conditioned medium, in fact, I think, so if you, my opinion, using conditioned medium from very well-defined cell line, let's say a cell line that produces a responding, I think that's perfectly fine. As far as you have your control and you know that it's only the responding or the growth factor what is doing the effect. Using conditioned medium from a cell line that you don't know what it is producing, I would not be in favor of that. But of course, if it allows your cell to grow and is the only thing, then it's maybe worth it to look what it is, the growth factor that is making the cells to grow. Regarding the stem cell cult from Stem Cell Technologies, I am aware that I've not tested it personally myself, but I'm aware that Hans Klev's lab has used it and it's working fantastic. So it is based exactly on the media composition that Toshi Sato in the lab described, and it is as good as using recombinant proteins.
Thank you. So another question is, how closely do the organoids resemble the in vivo culture system in terms of functionality? What you show is very impressive and close to an in vivo system, but what are the limitations of the organoid system that you have found?
Regarding the liver, for instance, I will just answer for the liver. Well, let's answer the different ones. So for the intestine, I think it's very close. Of course, the limitation is that you don't have a mesenchyme, so you cannot study mesenchyme epithelial interactions. For the functionality of the intestine is very close to the in vivo intestine, at least the epithelium part. And the same would be for the stomach. The same answer would count for the stomach. The liver is slightly different. The functionality of the hepatocyte-like cells that we obtain in culture is quite remarkably good, but it's not as it would be hepatocytes. So the human ones resemble, actually, surprisingly, the human ones resemble much better than the mouse ones to the functional hepatocytes. And I think that the difficulty will be to try to generate a culture system where you can maintain the full mature hepatocyte state for very long. My impression is that the cells that differentiate into hepatocytes, they die shortly after, and then we cannot maintain these cells. And I think that's a challenge.
Yeah, thank you. Another question is, do you think the DSS-induced pro-inflammatory environment affected the engraftment of the mini organoids? And have you been able to somewhat reproduce that without a DSS or injury model?
Oh, can you repeat the second part?
Have you been able to reproduce that engraftment without a DSS or injury model?
Okay, got it, yes. So absolutely, I do believe that the DSS caused this inflammatory response, and that this somehow attracted or facilitated the engraftment. I do think so, but I don't have any proof. And that I want to make it clear. And then what I do think is that I'm not aware that it has been tried to be reproduced without doing this damage, without giving the DSS. I would think would not be very efficient, or probably it will not even work, because I do think that if you want the cells to engraft, you need to have the space for them to home and engraft, because these are not tumoral cells. It would be surprising that they invade in a tissue where there is no lesion in the tissue. But that's, again, an hypothesis. I'm not aware the experiment has been done.
Okay, thank you. And then, have you ever tried to use organoids for bacterial infection models?
Yes, that's a very interesting question. Recently, a colleague in Hans Kleer's lab, she developed a human stomach organoid culture, and actually we did these experiments together, and we infected Helicobacter pylori into these stomach cultures. And what she observed is that there was an upregulation of pro-inflammatory, so there was a clear pro-inflammatory response after the bacterial infection. But infecting these cultures with bacteria, in that case, because the lumen is inside of the organoid, so the outside part of the organoid is the basal part of the cell, while the apical part of the cell is inside of the organoid. So it was a bit challenging because we had to inject the bacteria, literally inject the bacteria inside of the organoid. But it was possible, we did it, yeah.
Thank you. And then we'll have one last question, which is, this looks like a very powerful system. Have you tried, or have you been able to repopulate these cells with cells from the immune compartment associated with the epithelium in the intestine? And do you think it is achievable if you haven't?
We haven't, or at least I'm not aware of. And I do think it is achievable, yes. But I also do think that for that, it would be necessary to have mesenchymal attachment as well. But that's only my opinion. Yeah.
Yeah. Thank you. There are a few more questions, and what we'll do, some of them are culture related, so we'll send them out in the reminder email that we'll send out after the webinar, with also the recording of the webinar, and we'll answer them there. Because in the interest of time, we will conclude now. But thank you very much for giving us this talk today, Meritxell. It's been a pleasure to listen to all your exciting research, and it's been very captivating to follow your work over the past few years. Thanks again, everyone. Bye-bye.